This is part 2 of the lecture 5 on C. elegans. And so what we're going to talk about in particular today is to wrap up a little talk about the P granules. And then we're also going to talk a little bit about the step-by-step process for germline formation. But the big focus is on gut formation and C. elegans and predicting how certain mutations would affect this process. So we're looking at C. elegans, and we're going to take a look at the very initial steps at fertilization to see how molecules are asymmetrically localized and how this is going to set up axis formation for worms. And so the answer to this question is that yes, there are molecules that are asymmetrically localized at this early stage, which, by the way, this early stage is the one-cell stage just after fertilization. And just to talk you through this picture a little bit more, and then we'll talk about what's asymmetric. Basically, this is the single cell, and fertilization has just occurred. We can tell that due to the blue labels. So blue labels DNA, in particular the nuclei. You can see two nuclei. This is actually, in the blue on the left, is labeled as the oocyte. So this was the egg pronucleus, which is what they call it in a gamete. And then over here on the right, you can see a little bit of blue behind this red blob. That blue is the sperm pronuclei. So that's the name of the nucleus in the sperm prior to fertilization. And so basically fertilization has occurred. These two nuclei have not yet merged together, but basically what we see at this very initial step of fertilization, which you'll remember the sperm fertilizes at one site on the embryo, and that's going to help determine how the cells divide. And so what we see right at that point of fertilization is that all of a sudden all these little red dots here, which are all little tubulin proteins, they all flock over to the sperm pronucleus. So basically the sperm pronucleus organizes the tubulin. So basically, yes, at fertilization, tubulin flocks or moves toward the sperm pronucleus, and then this forms long microtubules. As a quick reminder about microtubules, these are one part of the cytoskeleton. Microtubules tend to be found within the cytoplasm, not right up against the edge of the cell membrane, but within, and they tend to move things around in the cell. And so basically, those microtubules are made up of tubulin proteins. So if these are each individual little tubulin proteins, which are actually shown in red over there, then they make these long rods. And so there are many little tubulin proteins, which make up these long rods, and this whole thing is a microtubule. So basically that's asymmetric. The tubulin is asymmetric once fertilization takes place due to the tubulin proteins starting to polymerize to form these microtubules. Now that we've seen that the microtubules are asymmetrical at that stage, we want to determine if that affects other molecules, and the answer is yes. In particular, it affects the P granules. They're shown in green on the far image. And P granules we talked about briefly previously. I'll tell you what they are now. So they are ribonucleoprotein complexes. This includes enzymes, RNAs, and other proteins. And basically, initially, here we see this is right when fertilization is taking place. We can see the pea granules are kind of punctate or diffuse across these little kind of polka dots across and throughout this cell. But once fertilization occurs, and then this is the event where the pronucleus of the oocyte and the sperm merge together, then all of a sudden these pea granules all get sucked over to the posterior side. And they're localized very tightly to that cell. So when cell division takes place along here, now we have, here's one cell and here's the second cell. And you should be able to figure out the name of that cell using the lineage in the earlier part of the PowerPoint that we talked about in class. So take a minute and find that and determine the name of the cell. I'm going to pause the video. And basically the pea granules are found at high levels in the P1 cell. So this is the P1 cell and this is the AB cell. And that means that anteriors on the left and posteriors on the right. You can assume that for these images. And just to write down the word that I mentioned for pea granules, so these are punctate, kind of polka dotted in their localization until the pronuclei merge. And basically remember that that's when these microtubules are localizing. And so basically they're going to start to grab onto these different pea granules and yank them over to this, to the posterior side. So my next question is, does the pea granule movement eventually affect the patterning of the adult? And the answer again is yes. The pea granules track with the germline. And in particular, they're a key molecule or kind of complex of molecules for germline development. This last part, about knowing that it's key for development, this would be experimentally determined. You can't just assume because something is found in a particular cell at a particular time that it has that role. And take a second and think about what kind of experiments you could do to determine that. Pause the video if you want. And so some of those experiments might be loss of function and gain of function. And just to talk you through this slide, again, we've got on the left-hand side, we've got the C. elegans embryo. This is where we left off on the last slide at the 2-cell stage, and then here we have the 4-cell stage. And if you look carefully at that 4-cell stage, this is the fluorescence. This is actually captured at the same time as this image and in the same cell. And so basically you can imagine overlaying these. And so basically imagine where this green fluorescence is in this embryo. And basically it overlays with the cell over here, which is the P2 cell. Okay, in the bottom part of this image, this is a whole adult worm, and basically in green, they're showing some cells or tissues that line up very tightly with this, and those are actually the adult worm gonads. So what's pretty cool is that the pea granules have an important role in their localization starting at fertilization, and that persists in the founder cell for the germline. And the pea granules are still present all the way through adulthood. That's not always the case for development. Sometimes there are certain molecules that are used early on, and then they kind of give rise to the next round of molecules and they themselves degrade or go on to do something else. In this case, the pea granules actually have a very persistent localization and presumably role in development. Okay, to summarize the axis formation in the early C. elegans embryo, basically first step is that the C. elegans egg breaks symmetry from having a sperm enter on a single side, and that's something we talked about during class time. And as you may recall, then what happens at that point was we had that array of microtubules. Sometimes, by the way, that's called an aster. Or spindle. And then the sperm pronucleus establishes polarity by creating this aster of microtubules. And in particular, then the important effect that that's going to have is that the aster microtubules are going to go grab onto P granules, and then they'll pull those to the posterior. So the aster microtubules will transport the P granules, and they go to the posterior most cell. And although we haven't talked about the details, so those P granules in turn contain some sort of signal that specifies the germline. You guys did this in class, so we're going to skip to the next slide. Okay, so that was to talk about germline and how that fate is specified. Again, really relying on the P granules. Don't forget to keep track of which cell name it is and what time in development we're talking about. Next thing we're going to talk about is how some other fates of lineages are specified, and in particular, we're going to really focus on the endoderm, which is the gut. And as we talked about before, this is a really nice system to choose because you can see that this E cell is the founder cell. This produces all of the gut cells in the whole worm, and then there's no other cell types that it produces. And just to kind of track through this lineage, so as a reminder, here are the cell names on the top. So 1 cell, C. elegans, 2 cell, 4 cell, 8 cell. And basically this one's sideways from the one we looked at before, but notice that the names of the cells are up here. So P0 lines up there. P0 divides into A, B, and P1, which we see here. It tells us the lineages later. And what we're really interested in is one of the offspring cells of P1, which P1 divides to produce EMS and P2. And so EMS is this cell down here. EMS is going to divide to produce two different cells, the E cell and the MS cell, which you can see in this location. And as we talked about previously, the E cell is going to produce the gut. You can think of that as the endoderm. So that's where the E comes from, endoderm. And then MS produces mesoderm fates. And that's where the MS name comes from. So I want to remind you a little bit about how we talked about different fate specification systems back in one of the early lectures. I think it was lecture 2. We talked specifically about conditional versus autonomous fate specification. And so take a minute to review that now. Pause the video if you like. And can you remember, or can you find out, which of those has to do with cell-cell interactions and cells signaling to each other? So that has to do with conditional fates. And so we're going to check out an experiment where they dissected out these different cells and then they put them next to each other to see who might be signaling to who and if conditional specification might be required for this EMS cell to produce E cell gut fates and or the MS fates, which produce mesoderm. Okay, so here's the experiments we're checking out here. Basically, they took a 4-cell stage embryo and then they pulled out different cells, so they dissected them out. Sounds tricky, and I'm sure it is, and requires some practice, like everything. And then they put different cells next to each other. And so basically, the first experiment that they did was they dissected out just the EMS cell and left it by itself to see what it would do on its own. without the rest of these 3 cells next to it. So the EMS cell, when it's by itself, produces 2 cells that have the MS fate. And so basically what that means is that there's lots of mesoderm that's produced, but there's no endoderm that's produced. So they knew that one of those cells needed to be talking to EMS to tell it to take on the endoderm fate. And so basically then they did some dissections to put the EMS cell, so they dissected out the EMS cell over here. They also dissected out the P2 cell and stuck them next to each other. And basically the result of that experiment here is that they got both the MS fate as well as the E fate, so that's back to normal. On the other hand, if they took the EMS cell and put it next to either the other 2 cells, either ABA or ABP, then they still only got mesoderm fates. And so think about what that means as a conclusion. And basically this is what it is, that the EMS cell receives some signal from P2 to produce the E cell fate. And so this is basically a summary that this is conditional specification. Saying that the conditions matter, that for the E fates, the endoderm fates, to be produced, then we need to have particular combinations that take place. So besides cutting things up and putting them in different orientations, the other thing that developmental biologists like to do when possible is to find mutants for whatever they're examining. And so that's what we're going to talk about here. And basically how they discover these mutants and create them is a long story, but basically I'll say that they found some mutants, and in particular they have a whole collection of mutants. I would say of maybe at least 5 to 10 different mutants that all produce more mesoderm, and they call all of these the mom mutants. And so basically we're looking at the fate lineage here. On the left we're looking at wild type, which of course is normal. We see again focusing on the 4-cell stage. On the other hand, in these more mesoderm mutants, and this would be a loss of function mutation, we always assume it's loss of function mutation when they say mutant, unless we hear otherwise. And so the mom mutants end up producing, they still have the same names of their cells, but notice that on the bottom here, the E cell produces mesoderm fate instead. And so mom itself stands for MO for more, and then the M for mesoderm, so mom. And you should definitely remember the names of these mutants as well as the fate changes. This slide is a little bit optional, but I think it helps you learn a little bit more about the sorts of studies that they can do and might give you some ideas for like your take-home quiz or maybe on the exam for sorts of experiments. So basically we're checking out a particular mutation which is called MOM2. And they numbered the different mom mutants. So each of those numbers indicates that a different gene has a loss of function mutation within it. And so basically what we're comparing here, so here we've got wild type, or WT. This is the MOM2 mutant, which is homozygous for this MOM2 loss of function mutation. And then on the far right over here, then this is a heterozygote. So this is a worm line which would have one good copy of MOM2, which is symbolized by the little plus sign. And then it's got a mutant for MOM2, which is this. So that's implying it's a mutation. And this one over here is MOM2 minus, MOM2 minus. So let's talk through the data that's here. So basically up here on the top, we see Nomarski, which is a type of microscopy. And this is a little fuzzy, but basically you can see that we've got, at the end of embryonic development, we've got this is the worm, which is wrapped around, it's still in the chorion. And then they stained this, this embryo or other ones like it for intestinal markers. So basically that's what this panel here in the middle is to look at intestine. And so basically if it's white, then that is a real bright white, then that's an indicator that the intestine has formed. And then they also looked at the pharynx, which is down here in the bottom. And so the pharynx, they basically could mark that or dissect it out, and you can see that there's a very clear delineated structure. So the pharynx does form. And so why this is significant is the pharynx is a mesoderm structure. And so basically the intestine does form. So the intestine forms in wild type, and then we see this pharynx mesoderm fate. So now let's compare that to the MOM2 homozygote mutant. And so basically, first off, this is just not really looking like much of a worm. There's not that nice clear line like we saw here. It's just kind of like a big amorphous blob as far as we can tell. Then if we look at the intestine, it's really this kind of diffuse backgroundy color. So this is not a really bright white. So basically the intestine doesn't form. And then when they dissect out the pharynx or stain for the pharynx, then check out that we've got this much bigger blob going on here. And granted, it's not like really well formed, but there's definitely a lot more of it there in terms of volume. And so this is where the name comes from. So again, this is more mesoderm. And then to get a better understanding about how many copies of this particular gene would be necessary, they also looked at the heterozygotes. You can see that these do not look great. So this does not perfectly match by any means the wild type. However, it definitely is has some more structure and might look a little more like a worm than this MOM2 mutant. And then again, we still see that there's quite a bit of mesoderm and not very much intestine that forms. So the two most important results on here are looking at the wild type and the MOM2 mutant. This heterozygote is just kind of an extra piece of information for us. And before we leave this, I want to sum up what the deal is with MOM2. So basically, what would we say based on this experiment? So is MOM2 necessary or sufficient? And what does it have to do with this fate formation? So take a second and come up with a conclusion about MOM2 and whether it's necessary or sufficient for a certain fate or a certain tissue to form. And so basically, the best way to go with this is to say that MOM2 is necessary because we're looking at a loss of function experiment here. That's why we use that word necessary. So MOM2 is necessary for endoderm formation. And another way to say that would be for intestinal formation. Remember that that's the opposite of what the name says. So the mutant name is more mesoderm. And so basically what that means is remember that it didn't produce. In these mutants, when MOM2 is gone, then there's lots of mesoderm, but no endoderm. And so that's why we say that MOM2 is necessary for the endoderm formation. We're going to skip this slide. So let's talk a little bit about what exactly is the role of MOM2. And so basically I'm going to kind of cut to the chase here and summarize a bunch of experiments and go back to this concept about the conditional specification and exactly what sorts of molecules are being involved here and what the gene names are for each of the products that's produced. So basically, let's take a look at this figure here. Again, we're at the 4-cell stage. And we talked about how P2 and EMS together would form the E and the MS cell fate. And now we've got some more details. So here we have the MOM2 label. So as it turns out, MOM2 needs to be produced by the P2 cell. And it's actually a ligand. So MOM2, the gene, encodes a ligand. And I'm just going to draw it like that. And that MOM2 ligand binds to a receptor that's called MOM5. The ligand, again, this is produced by the P2 cell, which is on the posterior side, and the MOM5 receptor is present on the EMS cell. And so we need both of these things to be present. We need both the receptor, MOM5, as well as the ligand, MOM2. And so both of those parts are the conditional signaling that takes place for the P2 cell to tell EMS to produce both mesoderm fates as well as endoderm fates. Moreover, I want to tie this into a downstream aspect of the signaling pathway. So you guys know that cell-cell signaling is typically going to result in a change in gene expression. And so basically, the MOM2 signal, before it affects gene expression, actually causes, as it says up here on the top, differential accumulation of a transcription factor, which is called POP1. And so basically, if we look down here at the bottom, the E cell is going to end up with low levels of POP1, but the MS cell is going to end up with high levels of POP1. Again, this is a transcription factor, and as you know, transcription factors are going to turn on a whole lot of different genes. And so basically the MS cell is going to turn on one collection of genes, but the the E cell is going to produce something different because it doesn't have very much of this POP1. So again, kind of off to the side, remember that POP1 is a transcription factor. This would be a specific transcription factor, by the way. And so I'd like you guys to think about what would you predict for a POP1 mutant? If there's a mutation in POP1, loss of function mutation, how would these fates be affected? So pause the video and think about that. So basically, if we have low levels of POP1, then that's going to mimic what's happening in the E cell. And so basically, for the POP1 mutation, there's actually more endoderm, and this is at the expense of mesoderm. And basically what that means is this is a mom and pop story. I had some fun saying that. I think the scientists had some fun coming up with that. I'm going to just go ahead and tell you, it took me a long time to understand why it was called POP1. I'm talking years. It just never dawned on me. So anyway, you guys should know the story about how these genes work together and how their products are going to work together for these fate signaling. And again, remember to keep track of the following. So keep track of molecules, what cells that they're located in, and then what tissue or tissues are produced. By the way, this figure also has a couple other molecules on it right here. This GLP1, APX1, just ignore those. We're not going to go into that. So basically know this process really well. Okay, and just to kind of take this and look at a summary figure checking out the C. elegans process of signaling. So up here at the top we've got fertilization occurring, 2-cell stage, 4-cell stage. Then we've got some color coding going on for the different germ layers. And then at the end, seeing that the endoderm, the E cell, has this yellow color and the mesoderm has a red color. And basically what I want to point out to you now is that this MOM2 and MOM5, cool names, have this very important role in C. elegans development to distinguish two different germ layers, which are crucial for their development and survival. These actually have counterparts or homologs in other organisms. So in C. elegans, the name is MOM2, but in other organisms, we use this word went, and so this includes in flies and mice and humans. So they have counterparts, and it's this ligand. Moreover, and this is the really crazy part, so in C. elegans, again, we've got our receptor here, which we call MOM5. It also has a homolog or counterpart, and in flies, it's called frizzled. And mice and humans also have a receptor protein. And so basically, this is the really cool part. This is the toolkit part that Sean Carroll was talking about in Endless Forms, which is to say that we have these tools, in this case, MOM2 and MOM5, which work really well together. And so they're used and conserved through development and through evolution in different organisms. And so they have different names, but they basically have the same function. We can really easily see the DNA and protein similarities across them. And so kind of the point of this is to say this is a conserved, evolutionarily conserved package or toolkit different cell fates. And again, right here, for C. elegans, this package, which we're calling MOM2 and MOM5, sets up the E versus MS fates. And it actually sets up different fates in other organisms. I'm not going to talk about those right now, but the important thing to see is that things that we thought at one point were just very specific and like, oh, this is just used for C. elegans and just for their E and MS cell fates, ends up being something that is shows up in other systems, and was one of the really big surprises in developmental biology when we started to sequence genes and understand this conservation. Skip this slide. And then this is a summary looking at the axis formation, so you can kind of run through these. We talked about the first ones early on, so I'm just going to kind of sum this up and remind you about those different types of specification, autonomous and conditional. So we talked about how the E and MS signaling has to do with conditional specification. And as it turns out, the P granules actually have to do with autonomous specification of the germline. And then this is conditional specification of endoderm versus mesoderm. And so notice that in the same organism, in C. elegans, we have two different processes taking place. Each particular process is going to be set for a particular structure or tissue that's being patterned. So basically the germline always uses P granules. It always, through experiments I didn't show you a whole lot about, it will always be necessary and potentially sufficient for germline fates. On the other hand, for this endoderm versus mesoderm fate, that's always conditional specification, and it always works with MOM2 and MOM5 and leads to this difference in the POP1 transcription factor.